Oh, we're live, everyone. Why do we have to be so early? Well, there are only so many hours in a day. I am currently in Australia, which means that basically it's 10 p.m. here at night. So I don't want to be any later. Also, there's basically the entire day that we have is we're live on YouTube. So someone's got to go early, and that's me. Are you seeing the live stream? Because I'm getting an error message on my end. Although I had this yesterday and it loaded just fine afterwards. No, I think we're all groovy. Okay, excellent. Okay, it's going to be action packed. I'm going to try and get through as much of this as I can. I'm going to go through monohybrid inheritance. I'm going to go through dihybrid inheritance. I'm also going to go through with you the key terms which underpin everything basically in A-level biology, which is why I'm going to start with them. It's not the sexiest, but it is probably the most important, that you understand how to recognize, how to use, and sometimes how to define those key terms, although you don't actually, you have to come up with the definitions of many of them. So you're going to need pen and paper. You're going to need a red, blue, green, and black pen. If you don't have those color pens right now, just bring them tomorrow. We can just make do. I'm going to be working off one of the template notes, which from my videos, my full course in the content guide. It's got no words basically written on it. In fact, I've got a title written on it and nothing else. So you basically have to begin with a plain piece of paper and create your notes along with me. Is everybody ready? In summary, this is going to basically be for everyone, all exam boards. I focus on AQA, OCR, and Excel. If you want more detail, if you want the rest of the course, it's go find it on tailoredtutors.co.uk. Okay. Excellent. Back for more, JLo. I think the templates might be in the description, actually, Mohammed. Yeah, I don't know. Rich Warburton, everything's a bit crazy right now. Rich Warburton actually set these up for me. So I think he may have copied the description from yesterday. Yeah, I think he did copy the description from yesterday. Okay, I'm going to have the chat vaguely visible. I'm not going to answer that many questions because it's going to be too slow if I do that. So let me share my iPad. No, that's record. I want to share. There is a massive delay on YouTube. It's just the way that it is. Okay, so inheritance key terms. Got the title in blue. If you're new to my color system, key terms are going to go in red. This is going to be a page of red, basically. I would start, if you've not printed out already, it's going to be too late because I'm starting right now. You can start from a blank piece of paper. And to be honest, you can, the templates will be useful for people watching after the live, watching the recording. I think during the live, just start with a plain piece of paper. Okay, I'm going to start with diploid. With just those of you who've just been in my AS class, what is a diploid organism? I defined it over there. I'm not going to wait around for an answer. So it's basically it's a cell or an organism with a pair of homologous chromosomes, one from each parent. So I mean, it's probably a slightly different definition to the one that I gave you earlier. So it's, and I'm going to say a cell. A pair, if I could write the word pair. Pair of homologous chromosomes, one from each parent. So therefore it has 2 alleles of each gene. Obviously, one from your mother, one from your father. What's next? Well, I've used the word gene. Let's go with gene. So a gene is a sequence of DNA that codes for a protein. You do, in fact, I'm going to star this one because sometimes they will ask you to define what that means and ask you to produce a definition, a sequence. That's what we're talking about when we mean a gene. So the next thing, we have an allele or allele, personal preference on your pronunciation of that. So this is a variation or a version of a gene. So, for example, let's chuck in an example. I do examples in green in my course. So you might be big T for tall plant, be a little t for dwarf. So they're both genes for plant height, but the different versions are big, tall, or dwarf. Okay, what's next? Genotype. So this is the combination of alleles or the genetic constitution of the organism. I'm going to say the combination of alleles in an organism. Alleles and genes, very confused, very often, really important to have a tight understanding of what they are to be able to use them as accurately as you can. Combination of alleles in an organism, I'm going to say. So, for example, if we have our big T and a little t, you could be homozygous dominant, you could be homozygous recessive, or you could be heterozygous. They are the three possible genotypes that you could have. And then the phenotypes. Phenotype, in fact, genotype, you might get asked if you're much more likely to get given phenotype as a definition. There's 2 marks for phenotype. What might you need to say? So it's I'm going to say the physical expression. So what you actually see of the genotype for 1 mark. What's the second mark? The observable characteristics would be fine. And what makes up the phenotype? The physical expression of the genotypes, of the genotype, and it's influenced by the environment. So 1 mark for genotype, 1 mark for the environment. You could say observable phenotype, the observable characteristics, the physical expression of, all of those things are not going to get you them. Well, they are the important bit to say it, but genotype and the environment is the first and second mark. So that is going to be the characteristic. So tall or dwarf in this case. Obviously, there will be some variation amongst the tall plants based upon good soil or bad soil. Yeah, so that some tall plants will be taller than some other tall plants because they had more nitrates in the soil. So there's some environmental variation, a range in that, but you would basically have a group of tall plants and you'd have a group of small plants and they'd have discontinuous variation between them. Okay, we've got dominant allele. Oh, I'm in green. And whilst I'm writing it, recessive. Dominant allele, recessive allele. Dominant allele is a dominant allele. Again, they could ask you to define this, and they could ask you to define this in terms of key terms. That's why I started with this. So it's an allele that's always expressed in the phenotype. An allele that, if it's present, is expressed in the phenotype. I'm going to say is always expressed in the phenotype. Recessive allele is only expressed if it's homozygous recessive. What would I say? An allele that is only expressed. When it's homozygous. So, for example, this in our case would be, let's do an e.g. little t, or it would be little t, which would give us a dwarf phenotype to the little t. There's also a codominant alleles. Domin. I need to spell them right for you. If you can read my writing on that, codominant. Allele. Okay. This is when both alleles are expressed as the phenotype is intermediate. So when both dominant alleles are expressed in the phenotype, I can't write dom in, that's dom. The phenotype is intermediate. So in between the two dominant alleles, it might be black fur, white fur, the codominant would have gray fur, in intermediate. All of this is going to be suitable to all exam boards. Homozygous, I have said, homozygous. So when both alleles are the same. Hetero. Zygous is when when alleles are not the same. Maybe give an example for each of these. E.g., well, that could be big T, or little t, and heterozygous would be like this. And gene locus is the location of a gene on a chromosome. If we were to look at my chromosome 12 and your chromosome 12 and Uncle Bob's chromosome 12, well, actually they're homologous pairs, there's two chromosome 12s, and you were to look at a specific locus on that chromosome, we'd all have the same gene. The allele could be different or would be different. Okay, you can come back to the recording, pause it at this point, and fill those in if you're not up to speed. But now I just want, they're important. Oh, I'm going backwards. Monohybrid inheritance, I want to try and fly through this as quickly as I can. This is relatively simple. So what is monohybrid inheritance? Let's define it first of all. You're going to let me, well, I didn't know I could do that. Are you going to let me draw on it at the same time? No. You're going to make it full screen and look pretty? No. Okay, so this is inheritance of a characteristic controlled by one gene. Mono, one gene. So inheritance of a characteristic or trait, characteristic controlled by a single gene. Only one gene. So for example, we could have our big T and our little t. In fact, let's do them separately and say what they actually stand for here. Tall plant, I'll do it. Dwarf plant. Okay, so I'm going to start with parent 1 and parent 2. Parent 1 I'm doing, generally in breeding experiments, you start with purebreds. So they are homozygous. Is if you see the word if you see purebred in the question stem, you can interpret that as being homozygous. So parent 1 is going to be homozygous dominant, and parent 2 is going to be homozygous recessive. This is generally where you begin breeding experiments. Nothing interesting is really going to happen here. But what gametes can parent 1 make? Well, you're either going to mean, you're going to gametes, this is diploid. We've got two T's, two big T's. In fact, let's put these in. Otherwise, you're going to, so we've got the parent genotype. And this is and then you've got the parents' gametes. Gametes are going to be haploid. So we're going to have one allele. Well, the only possible allele, we're going to have big T or big T. So there's basically T. In fact, I might write both of them just because, but basically it's big T or big T. And the little guy, well, we've got little t or we've got little t. So our gametes, I'm going to put them in circles. It's going to help clean things up. Okay, so what happens when we cross these things over? I'm going to put my little t's in here whilst I'm still in green. I'm going to put my big t's in here whilst it's still in blue. This would be our F1 cross. So we take our purebred homozygous parents and we do our F1 first filial generation. What happens? Can anyone tell me what the results of this is going to be? You take homozygous dominant crossed with homozygous recessive. You might be able to just visualize it here, but you're going to get always the same result every time you take a pure dominant with a pure recessive. You're going to get one big T and one little t of each. So they are going to be 100% heterozygous. And that's a little shortcut, but you can only do that. Same with dihybrid or polygenic if you're at Edexcel inheritance. If that's the case, then you will have 100% heterozygotes. I would generally avoid trying to do too many shortcuts in inheritance because it's quite easy to make mistakes or to make assumptions that you that might be wrong. So the genotype are all like this, and the phenotype are all tall. So to look at, they're all identical. And okay, so then we're going to self-cross. We're going to take these and inbreed them. So they are all going to be heterozygotes. This is why we start with pure homozygous, pure homozygous to get a pure heterozygous. And now the F2 generation is where the interesting stuff happens. And again, what will our F2 generation is going to be pure heterozygote. So I'm going to do parent 1. I'm just going to stick with blue for parent 1 as being like this. And parent 2, I'm just going to stick with being like this. So I've just got the two colors. So what are my genotype? In fact, I could just bring them down a little bit. Genotype and gametes. These are the things you need to know every single time. I've actually kind of done. I'm just going to move these. So the genotype would be this. The gamete, well, we can now have this gamete or we can have this gamete, the big T or the little t. And obviously these are the same. So we could have big T or we can have little t. So now let's populate our Punnett square. We have our big T here. So big T, big T, big T, little t, big T, little t, and then little t, little t. So our genotypes from the F2 cross, we're going to have big T, homozygous dominant times 1. We're going to have heterozygous times 2, and they're going to have homozygous recessive, one of these. Both of these are going to be tall, and this is going to be dwarf. So the ratio of tall to dwarf is 3 to 1, or you could maybe say 75% to 25%. Obviously, read the question carefully. If they say give it as a percentage. So a 3 to 1 ratio is characteristic, and that's how we get there. That's a relatively simple, obviously one gene going on, basically pretty straightforward. Dihybrid, I hear you asking. Here's the dihybrid. Wow, in fact, I'm ahead of schedule. You're doing a great job of answering each other's questions in the chat. If you can keep doing that, if there's sort of like issues you can't answer, then save them up. And I've got half an eye on it, but obviously I'm pretty much flat out writing stuff. Any little questions for me? Do you need to show the genotype next to the ratio? In the question, it was nearly always going to say, do the diagram and show, which means basically prove what the phenotypes are. Sometimes it asks you for the ratios, but it doesn't. Ratios, sometimes it asks you for and sometimes it doesn't. So I would say follow the question through, find some practice questions in the exam guide from us. You've got loads of examples of this in the inheritance section. Okay, so dihybrid inheritance. If you're at Excel, you'll call this polygenic inheritance. Polygenic or dihybrid. So it's inheritance of characteristics of, well, it's two characteristics that are controlled by separate genes. You would assume, unless the question tells you otherwise, they are on separate chromosomes. Because if you've got gene linkage going on, that's what I'm going to be covering tomorrow. You generally will assume that simple inheritance, it will tell you if they're on the same chromosome. So it's inheritance of two characteristics that are controlled by separate genes. Two genes. So this is what makes what's in the black box. Well, I've just pre-populated. Have I pre-populated the black box? I was planning to pre-populate the black box and cover it over. I'm not actually sure that I had time to pre-populate the black box. So we might see how it goes. Okay, so the parents' genotypes are, we've got homozygous dominant. So here I'm actually going to give you, let's go through these, e.g. we've still got our T as tall, and we've still got our little t as dwarf. I keep wanting to write it with a small d, dwarf. Now we're also going to have R for red flowers, and we're going to have a little r for yellow flowers. Okay, so we've got two different things, tall plant, dwarf plant, red flowers, yellow flowers. Two characteristics on two separate chromosomes. Okay, so parent one is going to be homozygous dominant, which means he's going to be little t, little r, little r. And parent one is going to be homozygous dominant, so big T, big R, big R. So what are the possible gametes? Now we've got a few more options, but there's not that many. We could have this big T with this big R. So we go, I'm in black. So we could, let's take away those underlines. We could have big T, big R. We could have the same T with the other R, which is going to give us the same. We could have the second T with the first R. What am I doing? Writing that. Or we could have the second T with the second R. So they're all four possible gametes are big T, big R. Well, we know this because they're homozygous. There's basically only one option each time. But I'm just sort of hammering this one home because it can be super confusing if you don't get it. So what is in the box is a very good question because, how do I delete the box? Is my next question. Boom! Very happy that I figured that, and I did fill it in. Look at me, TR. Okay, so I've just taken these guys and I've put them in here, and I've taken these guys and I've put them in here. And so we then do. You can see why I haven't. I'm going to draw out the next cross, and just filling in these boxes takes a little bit of time. So all our homozygous dominants are in here. So, and all our homozygous negatives in here. What do we get if we cross homozygous dominants with homozygous dominants? What do we get in the F1 generation? These are 100% heterozygotes. The genotype in this example is all big T, little t, big R, little r. And the phenotype are all tall and red flowers. Again, we're going to do the same. Now we're going to inbreed I see the word mistake. Is that something I've done? 16. Oh, no, okay. So again, I'm just going to go with, no, I'm in blue, big T, little t, big R, little r. I'm going to go green. I'm going to have difficulty fitting this in. Big T, big R, little r. So they're both the same. What gametes can we make? Now we've got a bit more selection in terms of gametes. We could have the big T with the big R. Oh, I'm in the wrong color again. We could have the big T with the little r. We could have the little t with the big R, or we could have the little t with the little r. So they're going to go in here. Big T, big R, big T, little r, little t, big R, if you can see that, and little t, little r. And obviously the other parent is the same. I'm just going to do them down the side here. I'm not going to draw them here. I'm just going to put them straight in here. Big T, big R. We could have the big T with the little r. We could have the little t with the big R, or we could have the little t with the little r. And we make equal proportions of these. Is there a mistake? If there's a mistake, try and describe where it is and I'll go fix it. Okay, so if we fill this out, I'm going to do we've got big T, big T's all the way down here. We've also got big R's all the way down here. And I'm just going to do 2 rows at a time, and little r's all the way down here. And then we have big T, little t, big R, little r, big R, little r, big T. Well, really the big R should come first. It doesn't matter in the cross at this point. Big T, little r, little t, big R, little t, little r. Oh, this word? Oh. No, looks right to me. Anyway. Let's go back. So we've then got, let's not do it. Let's leave a gap for the little t, 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 little t. We've got big R, big R, big R, big R, then little r, little r, little r, little r. Let's go back to green. Obviously, you won't have 2 separate colors in your exam. This is a big T and a big R. Big T and a little r. This is a little t and a big R and a little t and a little r. It's super easy to make mistakes when you're doing this quickly, but in the exam, you're going to be under time pressure as well. You don't usually have to draw them because it is quite time consuming. Okay. So now what have we got this? What have we got here? This is the F2 genotype or generation. And what have we got here? Well, we've got tall and red. We've got tall and yellow. We've got dwarf and red. And we've got dwarf and yellow. So anything with a big T is going to be tall, and anything with a big R is going to have red flowers. So I'm actually going to work my way backwards. So dwarf and yellow, what genotype would I need to give dwarf and yellow? Dwarf is controlled by homozygous dominant. And you're not answering the question that I'm asking. I'm asking what genotype do I need to be dwarf and yellow would have to be little t, little r, little r. Dwarf and red would have to be little t, and then either big R, and I'm just going to put X. It could be big R, could be big R, little r. I'm not going to put all the genotypes down. I'm actually going to put these. So for tall, big T X, big R X, and tall T and yellow would have to be little r, little r. X just meaning any possible one. Okay, so little t, we've got one of these. There's only one. I'm going to go away, page 1 of 1. I don't. Oh, it doesn't like me writing in the corner. Okay, so homozygous T's with any big R. So we've got one here, got one here, and we've got one here. The pattern is always the same if you lay this out in the same way. So we've got 3 of these. Now we're looking for big T's with little r's. So we've got one here, and one here. So there's also 3 of these. And that means everything else has either got one big T and one big R. And so there are 4 times 4 is 16. That means there are 9 remaining. So the phenotypic ratio that you would expect here is 9 to 3 to 3 to 1. Ratio. You would only expect this if they are on separate chromosomes. Do you want me just to show you kind of what, this is going a bit off grid, what will the difference if this was linked or if this was not linked? I haven't planned it, but it might be useful. This is when most students just get lost in autosomal linkage. Okay, we've got T's and R's. Okay. What the hell is going on here, Rich? Think this through. Okay, I am going to engage my brain here. This is going to be tricky. Okay, so this is our locus for T. Should I use the same colors? Let's use different colors. Okay, so this is going to be T. So this guy is, well, we started with our I think we want to start with heterozyte. It's not very easy for you to see that color, is it? I will maybe go with green. Okay, so if we start with heterozygotes, okay, so this is, oh. Okay. Okay, yeah. I think I'm getting there without making mistakes. Okay. So this is big T, big T. This is little r, little t, little t. Obviously, we've got homologous pairs. And that's where our T's, and they've duplicated. We've got the X-shaped. That's what's happening. And then we're going to go red. I'm going to do my big R, little r, little r. But here, they're on the same chromosome. So here we've got our big T, little t, little t. And here we've got our big R, big R, little r, little r. This is just the difference. So these are not linked, I suppose, not linked, and these are linked. So during meiosis now, what's going to happen? Well, first meiotic division, we are going to split these down here, split these down here. So we're going to end up with no, we're not. We're not splitting down the middle at all. We're splitting them down the middle here. Okay, so we'd end up with, I am winging this. I think it's going to be great. So this one's got our T's on it, big T's. So this one's got our little T's on it. This one's got our little R's on it, big R's on it. This one's got our little R's on it. And this one is big R, little R, little R, big T, little T, little T. Are you with me so far? This is meiosis 1. It's going to be a bit scrappy. So linked means that this is autosomal linkage on the right-hand side. I suppose we would call this autosomal because it's not on a sex chromosome. Autosomal, autosomally linked. And then our second division is going to split the centromeres. So now we're going to end up with 2 for that one, 2 for this one. Obviously, these are going to split down here, although, as I said, it would actually technically happen in the horizontal plane, if you were following my meiosis class from last time. So now I've got 1, 2, 1, 2, 1, 2, 1, 2, 1, 1, 1, 1. And here we would have big T, big R. These would have done some crossing over, so they wouldn't be genetically exactly the same. What am I doing? So the only possible gametes, okay, so what are the possible gametes that we can make if they are not on the same chromosomes? Okay, what I needed to have drawn to make this really effective. What happens during prophase of meiosis? To make my visual point here. Crossing over. Okay, so let's draw imaginary crossing over going on. Black. Between these guys. I knew there was something that I was missing from this diagram. So we then end up with this as a possible combination because you can just exchange these. Which means that we can end up with I'm not happy with this. Let's get rid of all this. I don't think we go backwards. Go to sleep at 4 in the morning. Well, or maybe I'll do some late night sessions. Would you like some sessions at 10 p.m.? that might be doable. And this is obviously meiosis 2. Okay, now I can understand where this diagram isn't making sense. Okay, so what I'm going to do is I'm going to copy. Can I just make the whole thing? Yeah, look at that, working digitally. Okay, I figured out why I'm not communicating what I want to communicate, and this is going to be perfect to finish on time. Wonderful. Okay, this is scenario 1. Scenario 2, alternative universe, Rick and Morty style. Okay, in a different world for not linked, also not linked. Forget crossing over. It's not helpful in this scenario. Due to independent assortment, which way they line up, it's possible for them to line up like this: the big T on the same side as the big R, and the little t on the same side as the little r. It's also possible, because they are independent of one another, they are on separate chromosomes, that you get this happening. So just I wonder if I can pinch this bit. No, but I can do this. Let's move this over a little bit, because I didn't plan this, and therefore all my spacings are a bit whack. Okay, let's just put this one through. So we have this and this in this alternative reality. So we have our big T, little t, little t. We have our little r, and big R, little r in this universe, which means that when we split these down the middle, so first we split down here, then we split down here. In this reality, we've got the big T, little t, little t, and then we have little r, little r, little r, big R, big R. So it's possible to have big T, little r, little t, big R, big T, big R, and little t, little r. These are the possible gametes that we can form, because they can line up during independent segregation separately. However, if they are linked over here, it's not possible for them to line up differently, because they're on the same chromosome. And therefore, there is no way of doing that shuffle. You've got one card, you can't shuffle it. If the T and the R on the same chromosome, means they are linked. They're autosomally linked, because it's an autosome. Autosomes are all of the chromosomes except the X and Y, the sex chromosomes. Does that now make sense? I knew I was struggling for where I was going wrong, but it's not possible to have two flipped scenarios when it's linked. They're on the same chromosome. You can't arrange them separately. Yes, in terms of their loci, if they're loci, but that's more to do with crossing over. It's the further apart they are on the same chromosome, the more likely they are to be separated during crossing over. 10:43 p.m. my time, 11:43 a.m. your time. Boom, nailed it. I'm going to stop that. I want some qualitative feedback. I also want more than 60 people here by the end of the week, although I'm actually not here Friday, so I'm here Monday through Thursday. What optimal, okay, so you were discussing times a day. What would be optimal time of day for you? I don't know if I'm going to be able to change things. To be honest, if we don't get numbers well over the 100, I don't know it's the best use of my time being here. I want this to be, I mean, it will probably take some time to pick up a bit of momentum, but I want loads of people here. And if loads of people are not here, then I'll do something else with my time, and I will find another way of helping out all those people who are studying at home. 11 a.m. perfect. Okay. With non-linked chromosomes, independent assortment means that you can either have the big T with the little t, with the little r, or the big R. So if this is my big, I mean, it's shocking. This is my big T, but obviously due to independent assortment, it's random as to whether the other gene is on a different chromosome. So you can just interchange the big with the small, which means it's possible to get the big T with the big R or the little r. Whereas if they're on the same chromosome, you can't do that shuffle. They're on the same chromosome. The only is there, okay, well, is there less genetic variation with linked chromosomes? If you're talking about those two particular characteristics, they are less likely to, they're more likely to be inherited together, basically, because the gametes are going to be the same. Okay, well, I can just share my screen again. And I do have to definitely finish on time because Ronnie needs the account to be broadcasting. Don't forget the study with me this afternoon if you want to do some more. I don't know where Zoom has gone. Okay, so if we look at, you don't end up with less variation really total because other genes will be on other chromosomes and they will be being shuffled. But if we look at what our parent was here, our parent was this, and our gametes down here are going to be basically this or this. We don't really get a choice here. We have this or this. And over here we have all four. The only exception to that is if crossing over does some crossing over things, it's possible to get a few numbers of what we call recombinants, which would be the big T with the big T with a little r and the little t with a big R. But these are going to be the vast majority. Most of it, crossing over is rare. It doesn't happen all the time. So these are gonna, and then when you self-cross this, you're basically, if you cross a big T, big R with a little t, little r, you're going to get, I'm not going to go down that road, actually. Just let's celebrate finishing on time. Okay, give me a quick score out of 10 for communicating that in terms of them. Yeah, I went quite quickly. I want to know if I pitched that about right. Give me, what about speed? Too fast, too slow, about right? I mean, obviously, I've got an hour a day or 45 minutes a day to spend with you. Generally, I'm getting okay to a little bit fast, which is probably exactly where I want to be, just pushing you a little bit. Obviously, the recordings are going to be here for you to come back and recap. So, okay, so maybe do 2 and slow down just a little bit. I'm going to be trying a whole bunch of different things. As I said, the course still exists. If you want to drill down into this, go into the course, go check it all out. Yeah, you can always rewatch the video. Exactly. Okay, great. That's me done for tonight. I will see you tomorrow for the autosomal linkage in a bit more detail, although I actually got to the crux of quite a lot of it there today. And what else will I do? Maybe some sex linkage, maybe chuck a question at you tomorrow. Maybe, yeah, do some stuff like that. Whew. Almost time for bed. I wish. I've still got a bazillion things to do before I go to bed, but yeah, take it easy. See you tomorrow.